This method can help answer key questions in the field of glomerular disease regarding the functional and structural phenotype of the glomerulus and allows for the mechanistic analysis of kidney pathology. The main advantage of this technique is that it is adaptable to all rodent models of glomerular disease.
For assessment of the urinary albumin-creatinine ratio, place one 6 to 8 week old male mouse into a metabolic mouse cage containing water and enrichment diet food in a quiet room. After 6 hours, return the mouse to their regular housing and collect a minimum of 50 microliters of the baseline urine sample from each cage, repeating the urine collection from once weekly to once monthly. At the experimental endpoint, harvest the kidneys from the abdomen of each animal and wash the organs in ice-cold PBS. To examine the cortical glomeruli, remove and dice one pole of the kidney cortex into 1 cubic millimeter pieces. To examine the deep juxtamedullary glomeruli, remove and dice a small section of the medulla into 1 cubic millimeter pieces. Then, place the fragments from each section of tissue into individual electron microscopy vials containing 5 milliliters of 2.5% glutaraldehyde solution and store the samples at 4 degrees Celsius. For histology of the cortical and juxtamedullary glomeruli, remove and fix the upper third of the kidney in 5 milliliters of 4% paraformaldehyde at 4 degrees Celsius.